of Department of Biotechnology welcomes you all for the international webinar series. The Department of Biotechnology, IILM Engineering, is one of the oldest department functioning since 2002. Our department witnessed presence of students from all over the country. The Department of Biotechnology follow contemporary teaching and research methodology and have very competitive faculties and subject specialists. Our college, IILM Engineering College, is AICTE approved and affiliated by Dr. APJ Abdul Kalam Technical University for both UG and PG programs. Our Department of Biotechnology is one of the 7 departments functioning in the college. The department has a state-of-the-art infrastructure and labs in the domain of genetic engineering, microbiology, fermentation technology, biochemistry, immunology, plant tissue culture, and molecular biology. The department has received several funded projects in last couple of years from the agencies such as Science and Engineering Research Board, VRPS, CSTUP, CRIP, etc. And it is one of the major department in Delhi, in NCR region, working with Unnat Bharat Abhiyan. Currently, the department is working on the area of several industrial problems. Now I request Dr. Nitin Garg to carry this program forward. Thank you.
Good morning. Welcome all the participants. Myself is Dr. Nitin Garg, Associate Professor in Department of Biotechnology, IILM CET Greater Noida. I welcome you all in this international webinar that is on application of genome-scale metabolic models to study microbe-microbe and host-virus interaction. I welcome Dr. Manish Kumar. Dr. Manish Kumar is a computational system biologist and working as a postdoctoral researcher in the Department of Pediatrics at University of California, San Diego, USA. He is currently involved in multiple projects to study marine and human microbiome ecosystems using multi-species and strains metabolic modeling. After securing the first position in MTech in biomineral processing from ISM Dhanbad, which is now IIT Dhanbad, he did his PhD in system biology in the Department of Chemical Engineering at IIT Gandhinagar. Before moving to the USA, he also worked as a researcher in the Department of Biology and Biological Engineering at the Chalmers University of Technology, Gothenburg, Sweden, in a project funded by Bill and Melinda Gates Foundation. He received several honors and grants such as Institute Research Fellowship 2010-15, IIT Gandhinagar, Young Presenter Award 2014, Clostridium 13, Shanghai, China, Overseas Research Experience Support 2014, IIT Gandhinagar, for visiting University of California, San Diego, DST International Travel Grant 2012, CSIR Foreign Travel Grant 2012, and MHRD Fellowship 2007 to 2009. He published more than 10 research papers and review in peer-reviewed journals, including Nature, Microbiology, and Metabolic Engineering, and 2 books chapter. He also presented his research work at various international conferences in Spain, UK, China, Sweden, Portugal, Denmark, and USA. I welcome Dr. Manish. Kindly start your presentation. Thank you all.
Thank you. Thank you very much, Dr. Garg, for this invitation and such a nice introduction. I also want to thank IILM to give me this opportunity to speak to, I think, so many people. I think we are almost 63 already. So, okay, I will go and start my today's talk. So today I will talk about, am I hearable? Can you hear me?
Yeah, you are audible.
Okay, so today I will talk about genome scale metabolic models. Mainly I will focus on the questions like, what are these models? What kind of biological questions these models can address, especially in understanding the microbe-microbe and host virus interactions. So basically, as Dr. Garg introduced me, like I am a computational biologist. I translate from organism metabolism to mathematical models and vice versa. In general, we metabolic modelers identify all components involved in metabolism, for example, genes, metabolites, reactions, and using this information along with how they are connected inside the metabolic network, we reconstruct entire metabolic networks. And basically in this process, we write this kind of S matrix that contains stoichiometric coefficient of all metabolites in corresponding reactions. And coefficients with negative sign represent reactants, and coefficient with positive sign denote products. So in this way, this matrix also contain the information how these metabolites and reactions are connecting to each other. One of the main advantage writing metabolic network in this mathematical format is that this matrix easily can be read by computers, and it makes our life super easy for running so many simulations in few minutes. So moreover, these models are based on some constraints and boundaries, as whole system should be mass balanced at steady state. Each reaction should be thermodynamically feasible, which defines direction of each reaction. Flux of each reaction should be between defined upper and lower bounds, which defines basically the capability of each enzyme. I don't want to bore you guys with the details of these details, but I want to highlight here is that we solve these metabolic models using linear programming optimization. And for this process, we need a biological objective function, which is in most cases is biomass production. And this biomass production is like a proxy for growth. It makes sense because primary objective of any cell is to grow in a given environment. And when I say solving these models, I meant for predicting flux of biomass production, which is a proxy for growth rate, as well as flux of each reaction in the model, which is proxy for rate of reaction. And further, if we see how we reconstruct this model, so in this reconstruction process, we start with the genome sequencing and annotation. Once we identify metabolic genes in the genome, we map these genes to reaction databases to assemble all relevant reactions. This assembly of reactions also called draft metabolic network reconstruction. If the targeted organism is eukaryotes, then we need to assign compartments like mitochondria, cytoplasm, and so on to each reaction. And each metabolic model has a biomass reaction, which is a proxy for the growth, which I mentioned before. We also need to add other metabolic reactions like transport reactions and exchange reactions. Exchange reactions are used to connect media metabolites to the internal metabolic network, and transport reaction define interconnectivities between different compartments. Because of the incomplete information, annotation information in genomes, in most of the cases, we have many gaps in network. And we need to fill these gaps, we need to add some reactions from the closely related organisms. For example, we developed these 58 human gut bacteria metabolic models, and we found many gaps in their genomes. So we added approximately 80% reactions to these model to fill these gaps. We need to perform more automatic, semi-automatic, and manual curations to crack several things in model, such as mass and charge balance, reaction directionality, gene reaction association. Next, we curate model for organism-specific phenotypes based on experimental data. For example, what these organisms consuming and produces. And finally, after this whole exercise, we have well curated functional metabolic model with us. If we visualize a metabolic model, this is what it looks like. This lights and edges nodes represented, and color of nodes different color of different nodes differentiates metabolites based on compartments. As you can see here, the size of these models and details these models incorporate provide a tremendous opportunity to explore in the metabolism of an organism. In this part of presentation, I would like to give you some examples to describe what kind of biological questions these models can address. One important application of these models is to predict growth rates under different growth conditions. Simulating the growth using a metabolic model is like how we grow cells in laboratory. We pour media into conical flasks and then inoculate cells to grow them. Similarly, in metabolic models, we use exchange reactions to connect media metabolized to extracellular space. And then we use transport reactions to connect these metabolites to the internal network. To illustrate the outcome of these simulations, I will use an example from a study where we reconstructed models of 58 human gut bacteria selected from 3 children groups, including healthy Swedish, healthy and malnourished Bangladeshi, and healthy and malnourished Malawian children. Here, you are seeing the taxonomic relationship between these 58 gut bacteria. So we use the models or these 58 models to predict growth rates for these bacteria. And we use, for this, we use 3 different media to grow, namely human breast milk, ready to use therapeutic food, and in silico rich media. In this triplot, you are seeing relative values of growth rates on all 3 media. Basically, metabolic models are data-driven models. Further, we can validate these predictions with experimental data. If predictions do not match to experimental data, we can go back and improve the model until model start generating biologically meaningful predictions. Another vital application of metabolic models is to predict the metabolic capabilities of microorganisms. Here, I want to highlight why models are critical tools to examine the metabolic capabilities, because not all bacteria are cultivable in laboratory. Metabolic models provide this advantage to predict what these bacteria can consume or produce without growing them in lab. Here, in this example, we predicted metabolic capability of gut bacteria from 3 children group, which I mentioned before, in the form of short chain fatty acid production and amino acid production. In results, we noticed that irrespective of health condition, gut microbiome in Swedish children illustrated more diverse metabolic capabilities compared to other 2 groups, which are Bangladeshi and Malawian children. These models are really good in providing such kind of hypotheses that can lead to further investigation, like we can check the dietary intake history of the children, and then we can go also check their disease history and so on. Metabolic models of a single species can also be used to predict growth capabilities of mutants. When we knock out the genes, we just can knock out in silico without doing any experimental work, and we can check whether they grow or not. And this particular exercise gives us the information whether this gene is essential for growth of the selected organism. So, for example, in this study, single gene knockout in TCA cycle were performed using E. coli model, and then each mutant model was grown on these 16 carbon sources. And bluish color here represent positive growth of mutant, and yellow color represent either these mutants were not growing or growing with very slow rate, which means these genes are essential for growth of E. coli. Further, in this study, these predictions were compared with experimental data, and more than 90% of these predictions were found in agreement with experimental measurements. Similarly, we can also use this model to perform in silico overexpression of any selected genes. As of now, I was talking about application of metabolic models of single species. Now we can see how these models can also be useful to explore microbial communities. So I assume everyone will be agreed with me when I say mostly microorganisms do not live in isolation in different natural environments. For example, we humans have more microbial cells than human cells in our body, and these microbes interact to each other and to host. Over the time, various experimental approaches have been developed to identify these microbes in a given microbial community. However, how these microbes interact to each other and to host is still difficult to address using experimental techniques. On the other hand, metabolic models demonstrated potential to address these questions up to some extent. I will start with the pairwise interactions between species. For decades, we know that microbes exchange metabolites while living in a community. We use information of these exchange metabolites to connect two metabolic models and optimize biomass of both models during growth simulation. And we applied this approach to our 58 models to analyze pairwise interactions. And these 58 models are the same which I just explained a few slides before. We have simulated all combination of two models in two different media. And we analyze the results looking the scenario like if both models growing faster, if one is growing faster than other, if both are growing slower, if both are not growing together, and so on. In microbial ecology, there are different terms for these all scenarios, which you can see here. I'm not going into details of these results, but I want to highlight here competition was found superior behavior to cooperation among different pair of microbes, irrespective which media we use to grow these and these bacteria are from which cohorts. So these light blue fractions in these pie charts represent the competitive behavior, and the green color denotes cooperation. Similarly, we have seen these a greater number of metabolites were involved in competition compared to cooperation. So reddish color represents higher number of metabolites, whereas bluish color represent lower number of metabolites. These results go consistent with the reports. Those suggested the competition is more stabilizing force in microbial communities. It is good to study pairwise interactions, especially for developing the tools for understanding microbial communities. However, in natural settings, microbes live in much complex communities. That makes it difficult to study the interaction between them. Modeling microbial communities is still a developing research topic, and some really nice approaches have been published recently. In one recent study, using metabolic models, hundreds of microbes from gut microbiome samples from Swedish and Danish individuals were grown together as a community to predict their growth rates. It was noticed that community growth did not vary significantly among different samples, which indicates that microbiome was capable to convert dietary substance effectively into overall microbial biomass. However, growth rate fluctuates significantly within individual genus. And it's possible to do such kind of in silico experiment through modeling, but still we don't have any technology to perform these experiments in experiment. That's why these particular advantages make these models very critical for producing hypotheses which can follow further experiments and validations. One more interesting topic I wanted to cover today: how metabolic models can be applied to explore the metabolic interactions between host and virus. Currently, I'm working on addressing these research questions in one of my projects. Using metabolic models, we can investigate how virus can affect the metabolism of host in order to replicate or increase the number of viral particles. As we all know, virus consume host resources for increasing the number of their copies. Mainly, these resources are amino acids and nucleic acids produced in host metabolism that are used to synthesize viral genome and viral proteins, respectively. For studying this host virus system, we incorporate viral protein and genome synthesizing reactions to the host metabolic model to make it a infected host model. And during simulation, we optimize biomass of host as well as the viral building blocks. For example, metabolic modeling were used to study a MS2 infected E. coli system. Basically, MS2 is a double-stranded RNA virus that can infect E. coli. Analyzing this infected host metabolic model, it was learned that during infection, the production of many amino acids and metabolites involved in pentose phosphate pathways was significantly upregulated compared to uninfected model. And this is an indication how virus infection can reprogram the host metabolism in its favor to replicate. I just wanted to give you the flavor how we can use this model to detect variation in metabolism of host while viral infections. I hope everyone is still awake. At this point, I would like to summarize my talk. I believe using several examples, I was able to convince you how metabolic models can be very useful tool to study metabolism of single species, as well as interactions between microbes in complex communities. We also saw how these models can be applied to understand host virus systems. And the another interesting area which I didn't cover today is integration of multi-omics data to metabolic models. This exercise will not only improve the prediction of models, but also help to build the models of complex microbial communities in the future. Now, I would like to thank all the people involved in the studies which I presented today. I also want to acknowledge funding agencies to provide financial support for our research work. Currently, I'm working with Professor Jangler in his research group at UCSD. And after all, I want to thank all of you for your attention. And if you have any questions, please do let me know. And in case you get some questions after this presentation, and right now you don't have anything to ask, so you can also write me on my email ID. And if you are social media person, you can just write me, just do the direct message on my Twitter ID. Thank you. Thank you very much.
Thank you, Manish. If anybody has any questions, then they can write the question in the chat box.
The question answer session is on. Participants, you may ask questions with Dr. Manish. So, sir, there is one question from my end. Sir, as you have discussed the models to predict several things, and it was really a very new subject for me. Although I am a plant virologist, but to using this models to predict so much of metabolic things is really really a new thing to me. Sir, you have used several models. Can we predict the cases of corona with the help of this model? I mean, India is a very complex system in terms of different races and different, you can understand what I'm trying to say. तो क्या सर यहाँ पे
Yeah.
predict कर सकते हैं?
So there is like some limitations with this modeling approach. First of all, like it only consider metabolism and all the metabolic genes, whether it is connected with host or with the virus. So it can easily predict like what kind of variation of metabolism happening in host or in viruses, but like predicting like this corona thing, I think it will be difficult. If you are talking about the testing point of view, I don't think so. This model can use like further detailed analysis once you know how they interact but how. And then this how question can be addressed with using these models. What kind of resources they are taking from the host and they are growing. And basically, when we write these viral reactions, they are based on sequences. So everything depends on like how detailed sequences we know about these viruses. And just the model can tell about the resources they use. So I don't think so there is like a direct answer for the COVID thing these model can give, but they can help in analyzing afterwards when we want to know details how they work, how this system works.
Okay, and sir, you have compared three cases, one from Bangladesh, one from some African country, I don't remember, and
Malawi,
one from
Malawi.
Yeah, Malawi, and one is from Sweden. So what exactly that comparison was?
Oh, so basically today I was focusing on genome scale models. Like I just wanted to give the flavor, what are these models and how they work. So I didn't go in a detail about this study. So basically we were trying to Bangladeshi children. And of course, in Sweden, we didn't have a data from the malnourished children. There is no data like this. But for Malawian children and Bangladeshi children, we had this data for healthy and malnourished children. So we just wanted to compare how their gut microbiota is changing in terms of microorganisms present in the gut microbiome, as well as about their functionality. And then we use this genome-scale metabolic model just to compare their metabolic functionality. So because we get this information, what kind of organisms, bacteria are present in different gut microbiota samples, and then these model can be used to check the functionality, metabolic functionality. So basically, in a summary, we wanted to compare the gut microbiota as like a snapshot, which we can do from the experiments also, as well as we wanted to compare the metabolic functionality of gut microbiota in these different children groups.
So, Dr. Nitin, no question from audience end?
No question from audience end, but I have a question. Can these type of microbe-microbe interaction with host virus is also
up to some extent, people are using these models. For example, we also have human metabolic models. People are improving it over the years. And then we have this detailed human metabolic based on, let's say, if you have this cancer in this particular tissue there. So we can make tissue specific models, and we can use the omics data from these cancerous cells and as well as the healthy cells. And we can constrain these models based on this data. So up to some extent, we can compare how metabolically these different healthy and cancerous tissue are functioning, but exactly we cannot predict like exact treatment or like real problem there. But still, these models can give us the clue how different these cells behaving after being cancerous.
Okay. Any other question from participants end?
I think there is no question. So,
Please on your video. Thank you all.
Department of Biotechnology, IILM Engineering, I extend a very hearty vote of thanks to Dr. Manish for gracing your important work and sharing with us your findings and opinions today. It was an excellent coverage of computational analysis of several metabolic processes. The topic of the model, which predict growth of individual members in the complex community, was a uh, real essence of this today's lecture. We are all inspired by your great words, sir. I extend my sincere thanks to Dr. Nitin and Dr. Garima for perfect logistic support for this occasion. I can't thank us beyond their comfort. So I thank you all. Thank you, participants. Thank you, sir, again.
Thank you very much.